All right, thank you for having me. Yeah, so I'm Robert Aboukhalil. Just to give you some context about my background, I did my undergrad in computer engineering and then did a PhD in biology at Cold Spring Harbor. And in terms of live site trainers, I'm not really that much of a trainer myself. I've given a couple workshops before. Probably my favorite was at Cold Spring Harbor. This is actually where I met Jason, was because we don't have undergrads. So we teach middle schoolers and high schoolers how to do things like DNA extraction and gel electrophoresis, which is quite fun for someone who came from a CS background, has no idea how to use a pipette. But in any case, I kind of wanted to talk to this group to get a sense for how sandbox.bio can be a useful tool for some of the workshops that you do or some of the tutorials you already have that you may want to bring to life, so to speak. So the reason why I built sandbox.bio was what I noticed is that whether you have tutorials in text or video or in-person workshops, there are two main challenges that I've noticed people having. One being that everybody needs to set up their own environment, or you need some sort of way to set up students' environments. And that's really challenging in bioinformatics, where some tools are just really painful to install. And the other thing is it can be scary to make mistakes. Like if you're doing a tutorial and they're storing temporary data in a folder, and then they want to delete that folder and you forget to put an underscore in a variable, and that just makes this variable empty, and then you accidentally wipe out all your files. That's kind of scary, and it's unrecoverable because there's no trash on the command line. And the other kind of mistake or the scary thing is having runaway cloud costs that are gonna cost you a ton of money, maybe because yourself or your students aren't super familiar with using the cloud in a way that doesn't result in runaway costs. Now, of course, it's the incentive on the cloud companies to make that as complicated as possible. So that's not surprising. And so where sandbox.bio comes in is it has bioinformatics tutorials that all run in the browser. So you have all these tools like SAMtools and Bowtie and Minimap, and they're all running inside of your browser. You don't have to set up anything to get started. So one example is, I was working with the Aaron Quinlan lab who made the BetTools tool, and we took their default tutorial that, it's a text tutorial, and we turned it into something like this, where the exact same text is on the left, but it's a bit more interactive, where if you come across a command, you can click on it and it gets executed in this command line environment. And just to specify, like, this is not a simulation. It's not like I say, if the user types BetTools, then return this. It's actually running the real BetTools, so I can go in and fiddle around with it in a way that's not really talked about in the tutorial, and you can do all sorts of stuff on your own to figure out what is the impact of tweaking parameters, which is like a very common way of learning. And so in this case, there's no environment to set up. You just load up the web page and everything runs in your browser. You can experiment as much as you want. You're not going to touch any of your files, and you're not going to spend a lot of money on cloud. We also have exercises for some of the tutorials, like the BetTools tutorial has exercises that we've broken down into smaller criteria, so you can see as you're completing the exercises, you get some sort of progress report, so you know you're on the right track to completing the exercise. The way these tutorials are defined are every step is just a markdown file, essentially. where the tutorial text lives. And then there are these special non-markdown front-end components, essentially, that let you say what command you want to allow users to enter, and this will turn into this button over here. And same thing with alerts, they just show up a little differently. And then once you have these markdown files for each step, you can kind of tie them all together with a configuration JSON file that contains some metadata about the tutorial, what it's called, who made it, as well as a bunch of steps and what these steps are called. And what's really neat about this is that you can also define a bunch of the data files that should come with the tutorial. So the users don't have to download it from somewhere else. Once they load the page, these files are available and are mounted on this virtual file system in the browser that they can use in the tutorial. And so that's primarily how we've been doing things. And this is one of the areas that I want to improve in the next version is, can we have like a UI where you can design these tutorials so that you don't have to do so much manual coding for these tutorials? So how it works under the hood, currently, every tool that we want to support, we need to compile it individually to WebAssembly. So in case you haven't heard of it, WebAssembly is this technology from 2017 that was built by all the browser vendors coming together and saying, look, folks, it's been 20 years and all we can do is run JavaScript, which has been really powerful, but how do we run more performant tools in the browser? How do we bring Google Earth, Photoshop, and these kinds of compute-heavy tools into the browser? And that's what WebAssembly is the answer for. Although they obviously didn't plan for this to be used for genomics, but it kind of fits quite well given that they are similar to a Google Earth or Photoshop. They're very compute intensive. And you want to be able to bring these genomics tools to the browser without rewriting everything in JavaScript. That would just be untenable. And so when a user goes to sandbox.bio, these WebAssembly modules are downloaded onto their browser. And then they can send commands directly to that module saying, give me bed tools intersect of such and such file, and it will return the answer immediately. So there's no communication with the server that's needed. And that's really nice, because the alternative architecture in a world where WebAssembly does not exist is, practically you would have to go to the server, and then I would have to manage a fleet of workers that would scale up and down, figure out how to execute some arbitrary user command, and then return that answer to the user every single time. And the reason why we use sandbox.bio is for the following reasons. At this point, sandbox.bio, because it uses WebAssembly, is essentially a static website. It's serving static assets. That's really cheap to host. It's super scalable. And I don't have to think very hard about security because everything runs in your browser. If this was running on my servers, I would have to make sure people can't just break out and start doing really anything. It's also more responsive because you don't need to go across the network every single time. And some of the alternatives to sandbox.bio, what they will do is they will actually spin up an entire container for every student. This makes it even more tricky when you have like bad internet, because when you're on the command line, any keystroke has to be sent to the server so that they can interpret it. Because if you're typing a command and you press tab to autocomplete, it will have to go to the server, figure out what am I auto-completing to, and then send it back to you. So that gets tricky real fast, and it requires you to have a very stable internet connection. Because if you drop it and then you can't communicate with that container, eventually they will just stop the container. And typically these other tools are paid. Sometimes they have some sort of free tier, but which makes sense because it's so expensive for them to maintain a whole container open for every single student. And so that gets tricky real fast. So there are quite a few limitations here that I'll talk about. The first two are not that bad of limitations. If you have a lot of data for a tutorial, it's probably best to subsample the data so you're not overwhelming the user's browser. And similarly, if you're using a lot of RAM, like if you're aligning some sequencing data to the human genome, that is probably going to take like 4 gigs of RAM. Most users, I think, do not want you to do that. So it's probably best to show the example on smaller genomes where you can more quickly iterate on the loading of the reference genome and on the alignment itself so that people see the answer more quickly. So these are limitations that we have workarounds for. The ones that we don't are, if I can't compile a tool to WebAssembly, I can't have a tutorial about it on sandbox.bio. So that has been a big limitation. And that is one thing I do want to address in V2, and I'll talk about how I want to do this. The other issue is, say you're using sandbox.bio for a tutorial, you're learning about these new awesome tools. How do you go from that to running real world analyses in your own hardware or on your own environment or HPC or what have you? And a lot of that, it's currently pretty hard to do. Like there's no clear path because with sandbox.bio, everything runs in the browser, and it's hard for students because they still have to learn how to install these tools. They're a bit more motivated now that they know that these tools are useful, but it's still a challenge. And so these two limitations are my primary focus for version 2 of sandbox.bio. So in other words, goals are, I want to support more bioinformatics tools. I want to make a smoother transition from the tutorial to the real world. And also I want to build more tools for trainers. Things like, can we have an easier interface for creating tutorials? Are there maybe tools that could be useful during the workshop? I'm not sure. I'm kind of curious to hear your thoughts on that. But the idea to solve number 1 is, instead of taking the approach of compiling every single tool to WebAssembly, we're going to turn to a project called V86. And so what they do is they let you run a whole operating system in the browser, like a full-blown Linux in the browser. So you can, instead of having to compile every tool to WebAssembly, I can compile it to normal binary, which is a lot easier. And then this is a CPU emulator that will take that end result and make it such that it can run in the browser. So this still uses WebAssembly because V86 itself, the engine is written in Rust, the simulator, and then it gets compiled to WebAssembly to allow it to be performant. And so I figured I would give you a very early preview of what this would look like. So here I'm running the Debian Linux operating system in the browser, and it kind of works just like your own computer. I can now do cool things like run top if I wanted to, which I previously could not in sandbox.bio because it's quite complex to compile to WebAssembly. More useful things like VI, I can now have a Vim tutorial. I've been meaning to have this for a very long time. And you could use all its features. You can search, go back and forth, close the file, and it just all works. You could also use less, which is also previously not possible. So this adds a whole bunch of tools that we can train folks on. And this is just the Linux tools side of things. I'm also able to install a lot more bioinformatics tools that I previously could not. So this opens up the way for adding a ton more, or supporting a ton of more tutorials. Now there are downsides. Because this is simulating a whole operating system, it is going to need to download about 50 megabytes of binary at the beginning. And then every time you call a command, it will download more for that command. And so the idea is that the tutorials will just initialize everything they need at first load, and then you won't have to download anything else from the internet. So this is a lot of data, but I think to me, I think it's worth this added initialization time if we can support a lot more tools in the browser. The other downside is that this v86 project does not support tools that need to run on 64-bit operating systems. It has to be able to compile it to a 32-bit CPU, essentially. So tools like vsearch and vg and mmseek would not be supported in v2. To be clear, they're also not supported in v1 either. So that's not really a big difference, but it does limit the amount of tools a little bit. But I think most bioinformatics tools do support compiling to 32-bit. And this is a feature that a lot of people want from v86. So my sense is that this will get implemented down the line, and then we can start supporting even more tools. For goal number 2, smoothing the transition, the amazing thing about v86 is that the way I set up this Debian environment is I created a Docker image where I install each tool by compiling it to binary, not WebAssembly. And then that gives me a Docker image. So if anybody wants to move from sandbox.bio to their own infrastructure, they can take this Docker image and run it anywhere they want. And so that means that the sandbox.bio environment will look exactly the same when they use it elsewhere. And so that gives us, for the first time, a much more smooth transition from learning about something in a tutorial and being able to apply it outside of the browser. I guess the third one, I talked about some ideas about having UIs to help trainers develop tutorials more quickly. But I'm curious to also hear from other folks about, what do you think about other tools that could be useful? What are your thoughts on maybe some of the initial downloads? And yeah, I'd love to have that conversation.